We're going to review briefly what we talked about last class, as usual, and we're going to talk about modes of speciation. That is, two different concepts of how species form. We're going to talk, because one of you asked me a question about how to appropriately cite figures in the term paper. Remember, the term paper is due 2 weeks from today, worth 20% of the points in the class, so full letter grade. If you don't turn it in on time, you'll definitely not get an A. So whatever you have done in 2 weeks, you better turn in 2 weeks. Then at the end, as I said last class, we'll talk about the wrap video project for anybody who wants to stick around. So hopefully we'll end about 35 minutes past the hour, leave about 10 or 15 minutes for discussion. 0 points, it's optional. If you don't want to hang around for that at that point, you can leave. Okay. Next time, we stop talking about species and we end talking about species. We'll get back to some questions about whether or not different human populations are different species or not. We'll talk about the out of Africa hypothesis, whether or not, which direction humans went when they left Africa, colonized the globe, relationships between human populations, and so forth. So that's usually a really interesting topic to discuss. One of the pre-zygotic reproductive isolation mechanisms we talked about last class was mechanical or sperm-egg contact issues. The idea that there are proteins on the sperm and proteins on the egg that have to recognize each other. They have to bind and interact positively together to allow sperm to actually penetrate the egg and fertilize. So I wanted to go through a little tiny bit more detail about that process and show one quick example. So all I showed last time was a cartoon. This time I get to actually show one slide that's got a tiny bit of data. So we're talking about gametic incompatibility. When you have members of two different species, sometimes those species are not able to produce hybrids together because their sperm and their eggs don't recognize each other as being from the same species. And what's important to know about this is that eggs, oocytes, are actually very protective of themselves. There are a number of barriers that a sperm has to overcome to be able to actually fertilize an oocyte. And so the sperm is packed with some contents here. The acrosomal membrane at the very head of the sperm has some proteases and other enzymes that allow it to try to bury into the outer coat of the egg to actually reach the membrane of the egg. So one thing that's important is that when the sperm contact the egg, that it be able to release these contents from the acrosomal process and try to actually get to the oocyte. And that's illustrated a little bit better. There are some interesting looking micrographs here on the bottom where somebody in vitro activated a sperm, made it think that it had contacted an oocyte, and you can see these different stages occurring. The sperm releasing acrosomal vesicle and its contents, and then some cytoskeletal membranes, microtubules, basically push the head of the sperm forward, and that's when the actual transfer of the nucleus from the sperm into the oocyte happens. And I did mention this concept last time. It's just another way of looking at it. That on the sperm, there's a protein called bindin, which is so cleverly named because it helps it bind to the oocyte. And there's also on the oocyte surface then, on the egg, there's a receptor protein for that bindin. So there's a bindin is a protein, there's a receptor on the oocyte that recognizes bindin, the protein that's on the surface of the sperm. And within a particular species, so in this case, what we're looking at is the bindin cartoons of bindin and its receptor from one species, in this case SP, which you don't have to know what it stands for. It's one species of sea urchin. And because sea urchins are really cool to study. And the bindin and the receptor from a different species, SF, on the right side. And you'll notice that as I cartooned last time, the authors of this have cartooned that there are particular surfaces that the bindin and its receptor have to interact with together to get positive binding. And what happens if you have a heterologous cross, shown here down on the bottom, if you have the sperm from one species, say SF, and the oocyte from SP, like on this side here, so an SF sperm, one species sperm, and the other species egg, those two proteins don't have the right shapes to fit together and to actually allow fertilization. Does anybody know why this is probably important in sea urchins? Why you would study this sort of process and how eggs and sperm interact with each other in sea urchins? Yeah, they broadcast spawn. Yeah, you're doing the right hand motion.
Don't know how to say it.
Yeah, so they're broadcast spawners. So sea urchins, the females just release their eggs and the males release their sperm, and they just float around in the water. So there's no mate recognition. There's no pre-zygotic isolation, that is, right before fertilization, that has to do with one sea urchin recognizing another sea urchin as a member of its own species or not. They just say, here, have some gametes. And they float around, and so there are a lot of, presumably, depending on where you are in the ocean and the time of the year, there are lots of sperm from different species of sea urchin floating around, trying to happen to run into, and they do happen to run into oocytes from lots of different species of sea urchin. And maybe your leg if you happen to be standing in the ocean. But that's why this process is studied in sea urchins, because presumably this process is absolutely critical for species-specific fertilization events. It's the only time when you make sure that you're fertilizing an oocyte or fertilizing a sperm of the same species is at this point of contact, when the sperm actually touches the oocyte. And that process actually happens right there. So after the sperm has touched the jelly coat, it releases those acrosomal membrane, those acrosomal contents, right? That digests that egg jelly layer, and that lets the sperm actually physically come in contact with the membrane of the oocyte, which is where those receptor proteins are. And if that process works effectively, then the sperm nucleus actually gets inside the oocyte, and you've got fertilization. So sea urchins are cool. Plus, some people think they're tasty too. One of the studies that was done on sea urchins was to figure out how specific are these interactions between the bindin proteins and their receptors in sea urchins. So what these scientists did was they took sulfated polysaccharides, which is another way of saying those egg, something about the egg coat. And they wanted to know if you added sperm from 3 different species of sea urchin, what percentage of the time would you actually get the acrosomal reaction to occur? That is, if you touch, as was shown in the previous micrographs, the photographs of the sperm, if you coat the sperm with these oocyte sulfated polysaccharides, what percentage of the time can you actually get those sperm to do the acrosomal reaction, to release the acrosomal contents, pretending like it's fertilizing the oocyte? So what's the trend here? So these are the oocyte types down here.
Same species. A higher percent.
Right, so every time you get the combination of the same species, sperm and egg, you get high frequency of acrosomal reaction. And in all of the other crosses you make between species, you basically measure no acrosomal reaction. That is, the sperm doesn't recognize the oocyte as a member of its own species. It doesn't release the acrosomal content. It doesn't try to fertilize the egg. So presumably, at least in sea urchins and in many other species as well, species-specific recognition of the sperm and the egg is really important. If you're not from the same species, you won't get fertilization. So that's one way that speciation can occur. Evolution of the shapes of these proteins, bindin and the bindin receptor. Any questions about all of that? So I just wanted to show another example. I mentioned this last class, hybrid male sterility, one of the two aspects of the biological species concept. Right. How do you define two members of the same species? Or two members of different species, rather? What's the biological species concept?
Being viable and fertile.
Right, yeah, right. So if they're actually or potentially interbreeding populations, if you cross them together and their hybrids are both viable and fertile, then you started with two members of the same species. So this harkens back to the three-spine stickleback movies we looked at last class. We had the Japan sea males that do the intense dorsal pricking display and try to stab their partners into mating with them, whatever. And the Japan sea males that are a little bit more gentle. And the same authors of that paper, Jun Catano and others who were working on this system and trying to understand, basically trying to understand this. So in this case, these are all images, 4 images of cross-sections of testes from stickleback males. Which stickleback male do you not want to be? 1, 2, 3, or 4. Yeah. There's something wrong with male number 4. He's got empty testes, no sperm. You don't have, I'm sure you don't know that in advance. Nobody's an expert other than June and maybe a few other people in what testes of three-spine stickleback fish look like. But 1, 2, and 3 are all packed full of mature sperm. Individual 4 is sterile. And individual 4 happens to be a hybrid. So J stands for Japan Sea. So this individual had a Japan Sea father, J. Father is usually written first in a genotype. And P had a Pacific Ocean female mother. So this is an F1 offspring from a Japan Sea by Pacific Ocean cross. No sperm, sterile hybrid. So that makes us think, what? Are the Japan Sea and the Pacific Ocean three-spine stickleback different species or the same species? Yeah, so if we're using the biological species concept and you make sterile hybrids, then these are two different species. Except, what about number 2? So now if you've got a Pacific Ocean mom and Pacific Ocean dad, father, and a Japan Sea mother, you're fine. This is bizarre, but this happens all the time in nature. Has anybody heard about this before? When you have hybrids and one sex is affected and not the other one?
I have heard for the cases of ligers and tigons that they're both very dependent on which one the father or mother is.
Right. So ligers being what, lion dads and tiger moms?
Yes.
You make ligers and tigons, or the tiger dad and the lion. Yeah. Yeah. That's why I love being a speciation geneticist. And then you get the bullshits, the bulldogs crossed to the shih tzus. Right, so this concept, and I'm only telling you this because I want you to have heard it at least once. You might have heard of this before. This won't be on the exam necessarily. This is called Haldane's rule. When you do 2 different crosses between 2 different populations or 2 different species, when only one sex from one cross direction is affected, that's Haldane's rule. And lots of people studying all sorts of different species, fruit flies, mice, humans, chimpanzees, stickleback fish, observe this pattern, butterflies, birds, that whenever there's going to be an affected individual in a hybrid cross, it's more often the individual, this is the specific detail of Haldane's rule, it's the individual that's the heterogametic sex. Heterogametic. Hopefully that rings a bell from genetics, although we talked about only briefly usually in genetics. So how do you define which sex is the heterogametic sex versus the homogametic sex? What does it mean to be heterogametic?
Isn't it 2 different sex chromosomes?
Yeah, it has to do with the sex chromosomes.
Homogametic is the same.
Right, so if you're homogametic, you have two X's, and the heterogametic sex is the one where the two sex chromosomes are different. So that's what Haldane's rule says, is if you're going to have hybrid problems, inviability or sterility, it's going to be occurring in the heterogametic sex, in this case males. And then there are other cases ZW, ZZ sex chromosome systems where the ZW individuals are females and ZZ sex chromosome individuals are males. And so in both of these cases, if you've got an XY sex chromosome system in your species that you're looking at, you expect sterility or inviability in males because they're the ones that have different sex chromosomes. But if you're dealing with birds, Lepidoptera, butterflies, moths, some snakes and lizards, you expect females to experience Haldane's rule, reduced viability or fertility in the female sex, not in the male sex. And nobody yet, there are some hypotheses about why Haldane's rule occurs. It's just called a rule and not a law because we don't have an answer to that question yet. We don't know what the mechanism is that causes specific inviability or infertility in just one sex.
So does that mean that female hybrids, like with XX, are still able to interbreed with other individuals and stuff?
That's usually the case, yes. That if you, for example, Japan Sea, Pacific Ocean females, they all produce oocytes that are fine.
Okay.
It's only the male hybrids, and in this case it was only in one cross direction. It was only the male hybrids that suffered from inviability or sterility. So that leads to the conclusion that in the stickleback case, maybe the Japan Sea and Pacific Ocean populations are on their way to becoming different species. They're just not all the way there yet. And we can in vitro fertilize, I used to be able to in vitro fertilize sticklebacks in the lab and get crosses to work. And this is an excellent point to remind you that one of the motivating, I don't know if this motivates you, but one of the motivating questions that I asked was about phylogenetics. And when you have a phylogenetic tree that shows one lineage splitting into two, what's happening right there? What processes, are there mutations going on? Are there geological events going on? What's happening when one population becomes two different species? And this is a great example of how this is sort of a gray area, where it can take a long time. We might be looking at sticklebacks here, where we've got hybrid male infertility or sterility in one cross direction, but not all the cross directions. So yet other processes might have to occur before we'd consider all possible combinations of crosses between Japan Sea, male and female, Pacific Ocean, male and female, all to be completely reproductively isolated, totally different species. So it's kind of it's almost a misnomer, it's almost misleading to draw phylogenetic trees with a specific point in time where it looks like one population just poof, became two. Because that process actually takes sometimes multiple steps. I don't know how you would draw a phylogenetic tree that wasn't misleading. But it's just easier to draw trees that actually branch as opposed to trees that have cloudy gray areas where the branches would be splitting.
I have a question.
Please.
In the previous slide for the post-zygotic one, 2
Yeah.
and 4, so with Haldane's rule, you said there's really no way to, we don't know the mechanism, so there's really no way we can predict which one is going to work and which one's not going to work in those cases, right? Because you have a male Pacific with a female from Japan and it worked, but yet you have a male from Japan and a female from Pacific and it didn't work. So how would we be able to determine that, or there's really no way, it's just
So we're still in the theory process in the evolutionary biology field of trying to come up with hypotheses to test about why Haldane's rule occurs.
Okay.
So yeah, you wouldn't be able to predict it necessarily. The only way to predict it, well, is to know something about the sex chromosomes of the species you're working with. And that's why this is a rule. So if you go into studying speciation in cats, say, cats have X and Y chromosomes. So if you were going to predict, if you cross two different populations of cats, if they're different species, which sex would be inviable or infertile?
The male.
The male, yeah. So this is just a trend, and we don't yet know the actual genetic or molecular details of why Haldane's rule occurs. Number of theories, but it's just an empirical trend that we observe. So this slide is a cartoon that's one model for how genetic changes in two different populations can cause speciation. It's called the Dobzhansky-Muller Incompatibility model, named after some famous geneticists. And the idea here is that you have a population long ago, so the ancestral genotype, there was a population of individuals, not just this one, and there are two genes we're looking at here that are important, A and B. And there is no genetic variation at locus A, that is, they both have this individual's homozygous. There's only one allele in every individual in the whole population. So everybody back here is A1 over A1, B1 over B1, a long time ago. So there's homogeneous population. And then that population splits into two, somehow. It doesn't matter how the population, either physically or by some other mechanism, that's what we're talking about after this slide. The population divides into two. So you can imagine that a mountain range separates these two populations. My crude attempt at drawing a mountain range could be my hair. And what happens after those two populations separate, in this case physically, although it doesn't have to be physical separation, what happens? We get a mutation that occurs in one population, because of course mutations happen all the time and they're random. So you can imagine that if you've got two different groups of organisms, one individual over there gets one mutation and another individual over here in the other population gets a second totally different mutation. So now we have some heterozygosity. In one population, now we have some heterozygotes. There's allele version 1 and allele A version 2. And the same process happens in the second population, but the mutation is in a different gene, gene B. And this is just a theory. It's been shown to actually occur in some species, but this is a theory, a model, if you will, for how speciation might occur. The problem is, and how this causes problems in hybrids, is that if these two populations come back together, down here, let's say one of these individuals happens to fly over the mountain range and mate with an individual from the other population. Now, for the first time in history, alleles A2 and B2 are found in the same individual. Because previously, allele 2, A2, had only ever been in the presence of the first version, B1, of gene B. So when A2 arose, when mutation happened, it occurred in the presence of A1 and B1. And when allele B2 showed up in nature for the very first time, it was only in individuals that had B1 and A1. So this genotype is fine, where you're heterozygous at B. And this genotype is fine, when you're only heterozygous at gene A. What's different about this individual? It's heterozygous at both loci. So this is the first time in history, in nature, there's been an individual of this species that's heterozygous for both of these mutations at the same time. And the theory is that being heterozygous at both loci causes a problem for the organism. So it's the combination of having those two new alleles together, A2 and B2, for the very first time, those gene products, the proteins that those genes produce, don't work properly together in the context of the organism. So A2 is transcribed, translated, produces an A2 version of whatever protein this is. And the B2 allele gets transcribed, translated, produces the B2 protein. And the A2 and B2 proteins do something bad to this organism.
Is there any way of knowing why it caused a problem?
Indeed. It takes a lot of work, though. And that's one thing that my lab is currently working on, a very similar process to this in C. briggsii, where we observe hybrids that are unfit. And all signs point to this sort of a process, where in the Indian population I study, there's a specific mutation that's fine. For example, let's call it A2. It's fine when there are no B2 alleles present in the same individual. So the Indian population is healthy. And in my Japanese population, there's a different mutation, B2, that's fine there. But when you make a hybrid between them and you combine A2 and B2, that causes dysfunction. In my case, there's biochemistry that we do to try to figure out what biochemical process. We're talking about protein-protein interactions. You might imagine that some process like ATP production doesn't work properly. Or this could be, what did we talk about earlier today that this could involve? Genetic changes in different populations that lead to inability to produce hybrids. So what could genes A and B be? What proteins could they produce that are important for reproduction? Right, so you could imagine, you could imagine that gene A is binding, and you could imagine that gene B is the receptor.
Okay, that was a perspective on
Good. Yes. I talk a lot about generalities, but yes, it's sometimes useful to go into details.
And would this be for any species, any across the board, this applies to any and all?
Yes. Yeah. Presumably this applies to any species that's diploid or more. So not haploid species of which, so we're not talking about bacteria. Sexually reproducing species, diploid or more, have at least 2 copies of every gene. Yeah. This sort of process could occur. It doesn't necessarily occur every time speciation happens. But this is a possible genetic basis of speciation. The real question is, how do you separate individuals from one population into 2 populations long enough that those 2 populations evolve to no longer recognize each other, be able to fertilize each other, their genitalia shapes change, something. So in other words, the question of how do populations diverge? How do you split one population into 2? We've talked about mountain ranges, rivers, geographical separation. So these next couple of slides are just to introduce or reinforce terms that you should have read about already, sympatry and allopatry. There's sympatric speciation, there's allopatric speciation. There's 2 broad categories of how species form. In sympatry, the 2 populations actually occur, and we've talked about this already, that's why this slide should look familiar. Sympatric speciation occurs when 2 populations exist in the same place at the same time, yet somehow the populations are separate. They're not physically separate, they're there, but there's no interbreeding, like that Dobzhansky-Muller model. You've got population A, you've got population B, but they're in the same place. So this was the example we used, the Hawthorne's Rhagoletis palmonella, the apple maggots, or apple flies, where they can occur in the same location in the same part of the world, but they prefer two different fruits. So they're reproductively active. These two populations of the same species are reproductively active at two different times during the season. When the hawthorn fruits for one population, and when the apple tree fruits for the second population. And that keeps those populations reproductively separate. That is, they don't have the opportunity to make hybrids over enough generations that those two populations turn into two different species. These are also called host races. The hosts being the apple tree and a hawthorn tree, and the races essentially being another word for population or species. So when one population diverges because they prefer different hosts, then you can have speciations occur. While the two populations of flies actually physically live in the same space. And sympatric speciation is interesting to evolutionary biologists because it's really hard to come up with these sorts of scenarios for how do you have a bunch of organisms living at the same place in the same time yet not breeding with each other. So temporal reproductive isolation like this is one of those methods. So my goal for your understanding of sympatry and allopatry is simply that you be able to apply those terms to a scenario. So if I tell you a scenario, if I describe a situation, could you tell me, does that sound more like sympatric speciation or allopatric speciation? And the main diagnostic character is, do the two populations live at the same place? Sympatry, same fatherlands, or do they live in two different places? Allo or other patry, allopatry. Which is usually a lot simpler to theorize because you can imagine, for example, in the stickleback fish we talked about, we've got fish that live in the Sea of Japan, and we've got fish that live out here in the Pacific Ocean. They live in two different places. They're allopatric. You've got some fish that live in the Sea of Japan, you've got some fish that live in the Pacific Ocean. And they only hybridize, that is, those two populations only interact with each other in nature where the Pacific Ocean meets the Sea of Japan. So there are only a couple really small areas in the world where hybridization could actually occur. In other words, what this means is that those Dobzhansky-Muller mutations, like allele A2, could occur in the Sea of Japan population, and that other mutation, B2, for example, might occur out in the Pacific Ocean. So you think of Japan as the mountain range that separates the two populations for a while. The Japan Sea population hangs out by itself, the Pacific Ocean out here, and they diverge genetically. They accumulate different mutations in the two different populations, and now, at the present time, if you give them the opportunity to mate with each other, you get sterile hybrids. That's allopatry. Two populations live in two different spots long enough to accumulate genetic changes that cause their hybrids to be unhealthy. Questions or concerns about this? Sometimes speciation occurs in sympatry, sometimes in allopatry. Most of the cases we have where we know how speciation occurs is this, allopatry. There are relatively few cases of sympatric speciation. This is the most, this here, and the next slide as well, last two things to cover for class. This is the most complicated version of the question I suggested I might ask you at some point. And so I'm not going to put this on an exam, I'm going to discuss it with you now. the concept of ring species. So for example, here we've got warblers, huge geographical ranges they occupy in the world. And in this case, we look at European and Asian warblers. They live in this whole area here that's highlighted in the different colors. They extend from the Baltic states, right, and Europe, across Asia, China, and then down into northern India, Tibet, the Himalayas. So they occupy a really large geographical range. And the shading represents hybridization, essentially. So you've got populations, you've got birds here that can mate with birds there, and birds there that mate with birds there. But of course, will you ever have a bird from Xi'an fly all the way to Europe and mate? In this case, not. You don't know anything about the migration of warblers, I didn't tell you. But they reproduce locally. But the point is that those birds can reproduce with those birds, and those birds with those birds, and those birds with those birds. So there's no physical separation. It's not population 1, population 2, with a mountain range in between. This is one huge continuous range of occupation by these warblers. So could you get speciation in this case? So you can imagine that birds that live a mile apart can probably mate with each other. And that bird can probably mate with that bird. And that bird could probably mate with this bird all across this entire range, hence the shading.
Should this be sympatry?
So, right, so does this count as sympatric? Because locally, all of the birds live in the same range. But then again, there are also members of this range that will never come in contact with each other because it's too far for them to fly. And that's the essence of the problem. Right, so is YK, if you cross YK and XN Xi'an together, Yekaterinburg, Russia, Xi'an, China, if you cross them together in the lab and they don't make viable fertile hybrids, is that allopatric speciation because those populations live in different places, or is it sympatric because they're connected by a gradient of members of the same species? And at what point then would you draw the line that says that's where we separate those two species, the YK species and the XN species, when those birds might be reproductively compatible with those and also reproductively compatible with those?
How similar are the YK and XN regions?
Great question, thank you. How closely related are the XN and the YK birds, warblers, from each other? So this is a typical prediction of a ring species, and of most genetics for that matter, when we're talking about speciation. If you plot how far apart two different populations are from each other, in kilometers in this case, and how genetically different they are, with 10 being more genetically different, you sequence the organisms, you ask how similar are their DNA, there's a linear correlation. So the farther physically distant two populations are, the more genetically different the two populations are. In other words, you might also consider that to be the probability, that axis to be the probability of being two different species. Because the more genetically different you are, the more likely you are not to be able to reproduce with another member of the same or different species. So if I was going to guess, I would say that if you've got a higher genetic distance, you're more likely to be two different species. And in this case, that correlates well with how far physically apart these birds live. And I'm sorry that Xi'an and Yekaterinburg aren't plotted on this, but I would guess that those are probably up here somewhere.
So would this particular example be allopatry or sympatry?
That's the question, and there is no answer. So what do you think?
That's why that's something I don't know.
So it's both, okay.
Maybe it's like sympatric for the closer ones, but allopatric for the farther ones.
Further apart, the more it is allopatry.
Sounds good to me. So if you're looking at birds that never come in contact, not actually potentially reproducing in nature, then you would call it sympatric. So that would be a fine conclusion. That these two birds, if they're reproductively incompatible, if they don't make viable fertile hybrids, that would be divergence that occurred in allopatry. But if you did happen to find that there was an example where two very closely related birds, physically closely related that is, were incompatible, that you might call that sympatry, or sympatric speciation. But we never see that in these warblers. So closely adjacent populations are always able to make hybrids, but there's a particular distance at which you no longer can make hybrids. So case in point, in that range that I just showed you, that's here on the left still, right, there are two populations. I didn't show Beijing here in the red area. Right, we've got Yekaterinburg, Xi'an, Beijing. Which, if any of these three pairs, you can make three different crosses, right? Xi'an, Yekaterinburg, Beijing, Yekaterinburg, or Beijing, Xi'an. You can do three possible different crosses with these three populations. Which one is most likely to create viable, fertile offspring? Beijing and Xi'an? Okay, except look at the map on the left, the range distribution. They're physically, geographically close together, but are they connected by warblers? That is, do warblers live here in the Himalayas in between them? No. So if a Xi'an bird was going to mate with a Beijing bird, it would actually have to go over there. Can't go across the Himalayas. So these are actually very, despite the fact that on the globe they're very physically proximal, they're actually very geographically separate. And in this case, the Beijing and Xi'an warblers are totally reproductively isolated. They live very close together physically in space, you know, as the crow flies, we would say, except warblers aren't crows, they can't fly across the Himalayas. So the Beijing and the Xi'an populations are two different species. They're the ends, if you think about it in this way, they're the ends of the ring. They're very physically far apart in range.
So would the top two be able to produce viable offspring?
So Yekaterinburg and Beijing. I don't know the answer, but I would suspect not. Again, because of the large genetic and physical distance between those two populations. Okay. So time to wrap up so that we can have a brief discussion about wrapping. Yeah. I just wanted to point out, this is why I put this slide in, that there is a ring species in California that's actually well known. So if anybody's interested in studying speciation and wants to work on some cool organisms, we've got that as well. Okay, oh, and finally, I'll do this quickly. So one of you did ask about how to cite figures that you might take from other sources if you want to put a graphic in your term paper. This is just an example from one from a previous semester. So they copied this figure from a published piece of literature, and in the caption that they wrote, which you should always write captions for figures you're putting in. So they wrote the caption here, right? They've labeled the figure number. So this is figure 1, the first figure from top to bottom that they put into their paper. And they've cited it here, Wertheim and Worobey 2009. It doesn't particularly matter which format of citation you use. APA, MLA, doesn't matter to me. But as long as you cite it. And then down here is an excerpt from their, down at the bottom here, excerpt from their references section that shows the full citation of Wertheim and Worobey. Okay. So they put a little tiny citation in the legend, the caption of the figure, and then the full citation is printed at the end of the paper in the references section. So that's all you have to do. So feel free to do, and if it's a website, cite the website. Right? The only time you get in trouble for plagiarizing is if you copy something and you don't tell me where you got it from. Any other questions? Okay. So, you're free to go. If you're interested in finding out what else we have to do with this wrap project, come down to the front, please. We'll let everybody else head out. Everybody have a fabulous weekend.